spreading infection, and then finally the systemic infection. What are biofilms? Bacteria can aggregate together and form biofilms comprising of cells embedded within a microbially produced matrix of extracellular polymeric substance. The steps which lead to the bacterial biofilm formation, as we know that the planktonic bacteria, they attach to the surface. Initially it is a reversible attachment, then it becomes irreversible attachment and cell to cell adhesion. Then there is proliferation of the bacteria. Then there is a maturation, and the secondary bacterial colony also joins the biofilm, and then finally the biofilm collapses and the cells are released. See, the biofilm structure varies according to the microbial species and their motility characteristics. It can appear as a flat biofilm with only a few layers, microcolonies, stalks, and multi-layer mushroom-like structures, as you seen earlier. More complex structures frequently have water channels incorporated to allow liquid and oxygen flow, improving nutrient transport into cells, but also aid the delivery of antimicrobials to inner biofilm layers. The population in biofilms is diverse and includes cells at different growth stages and also into antimicrobial resistant and persister cells. Quorum sensing quality of bacteria changes the expression and regulation of genes, virulence, microbial competence, and antibiotic resistance of the cells. It limits the leukocyte and bactericidal product penetration and avoiding cell phagocytosis. The are, phages are small viral entities that infect specific strains of bacteria. Since they infect the specific strains, also known as the biological bullets, and they are abundant on our planet. They were discovered independently by Frederick William Pott in 1915 and Felix de Herel in 1917. They have been used for the treatment of infection in Eastern European countries and the former Soviet Union from 1920 to 30. And at that time, the indication for usage of phages was the treatment of dysentery, cholera, plague, skin infections including wound infection, respiratory tract infection. Phages were safe and effective in patients with dysentery and plague. French article in 1921 described injection into and around the skin infections, eliminating the infection within 24 to 48 hours. Now, how to isolate these bacteriophages? There are various steps which are involved in the isolation of the phages, and this was carried out in the microbiology department by the microbiologist. So there's a collection of water samples, removal of the contaminants, then sensitization of the bacteriophage, plaque formation, then the harvesting of the bacteriophage, bulk production, sensitivity analysis, then the polyethylene glycol precipitation to remove the endotoxins, and then the final storage for future use. Phage therapy relies on the isolation and use of phages that exist naturally in the environment. Phages are screened against frequently occurring pathogens, including drug-resistant and MDR bacteria, and evaluated through in vitro and in vivo models. Due to phages' high specificity for their bacterial host, phage cocktail formulations usually guarantee a broader spectrum of activity. Use of these mixtures targeting various receptors decreases the likelihood of the emergence of phage-resistant bacterial mutants. Phage formulations can be applied alone or in combination with other antimicrobial agents, and their administration is possible using various routes like parenteral, oral, or local. Are there any reference institutions for the phage therapy? The answer is yes. The Eliava Institute of Bacteriophages, Microbiology and Virology, established in 1923 in Tbilisi, Georgia. Ludwig Hirschfeld Institute of Immunology and Experimental Therapy, established in 1952 in Wrocław, Poland. Recent studies have demonstrated the success of phage therapy in ex vivo and in vivo animal models and even in human patients. Phage therapy in animal models such as in rodents and pigs showed antimicrobial potential and wound healing capacity after the establishment of an infection with Staph aureus, Pseudomonas aeruginosa, and Acinetobacter baumannii. So we conducted an in vivo wound model utilizing bacteriophage therapy of Pseudomonas aeruginosa biofilm. We created a subcutaneous pouch on the back of the 24 albino mice And these mice were divided into 4 groups. Group A, B, C, D. The phage sensitivity was tested, and phage cocktail solution was used after implanting the two phage, the bacteria latent catheters in group C and D, and a sterile catheter was implanted in the group B, and the only subcutaneous pouch was treated in group A. The group D received 10 microliter of 10 to the power 6 phages forming units per ml for 10 days, and group B and C received 10 microliter of normal saline. After 10 days, the catheter sections were explanted from group B, C, and D, and the tissue biopsy was taken from group A pockets and culture of bacteria and the phage colony counts. A significant drop in bacterial counts from 3.87 into 10 to the power 6 to 3.52 into 10 to the power 4 was observed in group D when compared with group C, and the difference was statistically significant. A significant rise in the phage counts from 1 into 10 to the power 7 to 6.81 into 10 to the power 8 was also observed in group D when compared with the baseline counts, and the difference was statistically significant. And this indicated the phage proliferation and successful bacterial kill in group D. So the present laboratory study could be indicative of a new treatment approach by multi-drug resistant bacterial infection, including wound infections. The phage therapy has been permitted for the treatment of bacterial infections in Queen Astrid Military Hospital in Brussels, Belgium in 2007. Requests for phage therapy in this hospital has significantly increased since 2018. Rhodes et al. Reported in 2009 the phase 1 clinical trial using phage therapy in chronic venous leg ulcers of 42 patients treated with Pseudomonas aeruginosa aureus and the E. coli cocktail. The topical phage application successfully treated these Staph aureus infected ulcerations. Another study used biobacteriophage preparation for the treatment of diabetic foot ulcer in 2 MRSA colonized patients that healed and showed no signs of MRSA. Morozova et al. They also reported there is no side effect of this adverse effect of this phage therapy. We conducted a trial of phage therapy in 20 patients aged 12 to 60 years of age with chronic non-healing wounds. These wounds presented with E. coli, Staph aureus, and Pseudomonas aeruginosa, and 3 to 5 doses of the phage therapy led to complete healing or healthy granulation tissue in these wounds. And we concluded the topical bacteriophage application may be quite effective therapy for the treatment of chronic non-healing wounds. Now, question is, should we use the individual phages or the cocktails? Susceptibility changes to the phages used can be due to the downregulation, shielding, or modification of the host receptors that are essential for viral attachments, or switching from a lysogenic to lytic cycle. Phenotypic variations can be solved by combining different phages in a cocktail. As you see here, a lytic cycle, what happens is the phage attaches to the cell receptor, then it enters into the cell, then it proliferates within the cell, and then the cell ruptures and these phages are released to kill the bacteria. But if you look at the lysogenic cycle, the basic difference between lytic and lysogenic cycle is that in lysogenic, the phage attaches to the cell receptor, it enters into the cell, and then it enters into the DNA, inserts into the host chromosome, and then the host divides, the phages proliferate, and then the cell ruptures and they are released to kill the bacteria. So a phage cocktail formulation should be from a natural source without genetic manipulation, should consist of only lytic phages, active against a broad range of target bacteria, able to replicate on the target bacteria and have high rates of adsorption, a short latent period, and a large burst size. Consists of phages with different bacterial cell wall receptors, the recognition sites to overcome potential bacterial resistance mechanism, capable of maintaining the killing ability throughout treatment and storage. What are the various routes of administration of phages? It can be parenteral, that is IM, intravenous or intraperitoneal, oral or local phage delivery. Lytic phages within topical solutions such as ointments, creams, and lotions can enhance the healing process of wounds. The phage delivery systems can be of different types. As you can see in this slide, it can be hydrogel, it can be used with the fibers, films, adhesins, nanospheres, liposomes, and so on. We evaluated the phage therapy in chronic wounds of 45 patients, 37 healed, and the healing rate was about 82%. Out of these 45, 19 were non-diabetic and 26 were diabetic, and the healing was equally good in both. This is just to show you a 55-year-old diabetic male presenting with 10 weeks duration of a right grade 2 ulcer, treated with customized phage therapy, and you can see the healing in 90 days. Another diabetic patient of 52 years age with a non-healing wound of 130 weeks duration, treated with phage therapy, and this healed in 90 days. Another patient with diabetic foot ulcer, you can see the ulcer on the heel, treated with the phage therapy, and you can see the wound has reduced in size by 75 days. This is another patient with diabetic foot ulcer, a plantar ulcer, treated with phage therapy and healed within 90 days. Again, diabetic foot ulcer, treated with phage therapy. You can see in 60 days the wound has reduced in size. Again, diabetic patient presented with the leg ulcer, treated with phage therapy, healed in 90 days. So we concluded the customized local phage therapy is very promising in non-healing ulcers. So to conclude, Mr. Chairman, ladies and gentlemen, non-healing wounds are an increasing health and global financial burden and are challenging to treat due to biofilm forming and MDR bacteria. Phages have proved to have an active bactericidal activity even against antibiotic-resistant bacteria and are a promising alternative to conventional antibiotics. Thank you. Jai Hind.